photosynthesis is mainly divided into two sections, the light-dependent reactions, and today we're going to start talking about the light-independent reactions. When we look at photosynthesis, we're keeping in mind that we are trying to capture light energy and transform it into chemical energy in the form of an organic compound. And so photosynthesis, the basic equation is CO2 plus H2O plus light produces glucose, C6H12O6 plus O2. And of course you can balance this equation by adding your coefficients of 6 here and here. When we look at the light-dependent reactions, we have mainly seen the use of light and water and the release of oxygen, but we have not used carbon dioxide. So today when we talk about the light-independent reactions, we will be actually fixing carbon dioxide or taking CO2 and putting it into an organic form. Before we go any further, we do need to talk a little bit about the structure of where this is actually happening. So the majority of photosynthesis is going to occur in the leaves. Usually anywhere where you see green, photosynthesis is occurring because it has a chloroplast with chlorophyll in it. Keeping in mind chlorophyll is what reflects the green light, and so that is what makes parts of the plant appear green to us. But not all parts of the plant are going to have chloroplasts and chlorophyll. A flower's function is very different. It's there for reproduction. It's not there to capture light energy. It's actually there to attract animals. The root's function is very different also. It's there to absorb water. It's not getting any light, so there is no need to create those organelles, create those proteins, because they're not going to be used. The majority of photosynthesis is going to occur in the leaves. We need to do a little bit of leaf anatomy. The epidermis of the leaf is going to have a waxy coating. And this is going to repel water, meaning that water does not get absorbed by the leaves. It's actually why leaves are always tilted a little bit. They're not perfectly flat, because when the leaf is tilted, the water will flow down into the soil where it can be absorbed by the roots. What we're interested in the leaf is this middle part. And when you feel a leaf, it doesn't feel usually like there's a lot in the middle, but there's a layer of cells called the mesophyll. And the mesophyll is going to be the majority of the photosynthetic cells. So we've got a palisade and a spongy mesophyll, but this is going to be where most of the photosynthesis occurs, in these layers called the mesophyll. The interesting thing about the spongy mesophyll is that there are spaces between the cells. So we can see the cells are not as tightly connected as in the palisade mesophyll. And these spaces are important for gas exchange. The gases can wait here in between the cells. There's space for the gas. And so how do plants exchange gases? The majority of the consumption of gas in a plant, so what gas does the plant use most, is going to be CO2. So we're going to see CO2 is mainly going to be going into the plant body, and oxygen is mainly going to be coming out. Does this mean plants don't use oxygen at all? No. They definitely use oxygen. But they're going to be using more carbon dioxide and releasing more oxygen in comparison to each other. So how do they do that? They don't have a nose or a mouth. They don't have those tubes like we do, but they do actually have small openings here at the bottom. And so this opening is called a stomata. And that's what the opening or the pore is called. And that opening is actually created by cells called guard cells. So the guard cells will actually open or close to regulate this opening, called the stomata. And these are usually found in the bottom layer of the cell. We don't see a lot of stomata up here at the top. The other thing that we need to add is that sometimes escaping through that hole is actually water vapor. We can't see it, but actually if you put a bag over a leaf, the bag will actually collect some water that's escaping from the plant. And the escape of that water actually is what provides part of the tension to pull water up the stem, along with cohesion and adhesion. So what's important about the leaf anatomy is that we have these layers that aren't doing any photosynthesis. They're there for protection. Then we have these layers of cells that are doing photosynthesis. But we also have these little openings at the bottom to allow for gas exchange. And we know that oxygen is going to be created, O2 is going to be created during the light-dependent reaction. So let's just take a second and look back at that. Inside of the chloroplast are these stacks of membranes called, the stacks are called granum. And each individual membrane is called a thylakoid membrane. Inside of the thylakoid membrane is a compartment that we call the thylakoid space. And the external area is called the stroma. And so during the light-dependent reactions, light was absorbed by proteins at photosystem 2 first. That light helped split water and we released oxygen. This oxygen will actually diffuse out of the chloroplast into the cytoplasm and then out into the space between the spongy mesophyll cells. So we can actually track the diffusion of oxygen out. The hydrogen ions cannot diffuse. The hydrogen ions from the water are stuck here in the thylakoid space. The energy from the light actually excited the electrons from splitting water. That electron was first used to help pump more hydrogen ions in, keeping in mind that we're using the energy from the electron to do the pumping. So again, what we see here is this hydrogen ion gradient. And so we have an electrochemical gradient. Electrochemical gradient, that hydrogen ion gradient. And as those hydrogen ions flow through ATP synthase, so as they diffuse, as they move from a high concentration to a low, we're going to see phosphorylation. And it's still called chemiosmosis, but it's not called oxidative phosphorylation anymore. Now it's called photophosphorylation. So we've made ATP. We've used energy from the sun to actually make ATP. Another input of light is used to excite the electron again before we pass that high energy electron to a carrier. And so at the end of the light-dependent reactions, we have a high energy electron being carried, and we have a source of ATP energy as well. And so what we're looking at today is going to something called the Calvin cycle, and this is going to occur in the stroma. So we're already there in the stroma. ATP is made out here. NADP plus is reduced in the stroma. So we're already in the stroma, and the Calvin cycle is going to be where we fix CO2 into an organic form. So that's what we're going to look at next, is what's called the light-independent reactions. It used to be called the dark reactions, but it was actually discovered that a lot of the enzymes that are used here are light-activated. So even light-independent reactions isn't quite the best word for it, because without light, a lot of the enzymes needed here will not be activated. So the question is now that we're keeping in mind that we're trying to build glucose. We're trying to build an organic carbohydrate. And so how do we use ATP and NADPH to build an organic molecule? What we're going to end up using is 3 carbon dioxides, and we're going to attach them to an intermediate. So if you just glance at the cycle here, it does look very similar to the Krebs cycle, except instead of slowly pulling electrons and carbons off, we're actually slowly adding electrons and carbons. The Calvin cycle here is very summarized, so there are a lot of steps here that aren't being shown because all that you're expected to know is the general overview of what's happening. So what is happening? We have a 5 carbon compound. 5 carbon RUBP, ribulose bis phosphate. So a 5 carbon compound is given a 1 carbon dioxide to become a 6 carbon. But that 6 carbon is actually immediately broken into 3 carbons. So the smallest unit would be 1 ribulose bisphosphate, 1 CO2, 2, this is going to be PGA, which we don't need to know. But what actually happens is that this has to happen 3 times simultaneously. And so we actually need 3 ribulose bisphosphates, 3 CO2s, and we get 6. This is PGA. So if we kind of think about that, 3 times 5 is really 15. So we've really got 15 carbons plus 3 carbons. And we end up with 18 carbons, but they're split into 6 threes. So it's a little bit tricky here. The problem is that this enzyme is only going to really work 3 at a time. And here is probably the most important part of the Calvin cycle, is this enzyme Rubisco. Rubisco is the name of the enzyme, and it is going to fix CO2. So it is going to act as a carboxylase. It is going to add carbon on. And so we have our intermediate, we add our carbon on, and we have these smaller molecules. What we do next is actually add phosphates on. This is going to change the shape. So really all that we're doing here is adding this ATP, this energy is helping to change the shape. We need that energy to change the shape so that it can fit with the next enzymes. What happens next is the idea of reduction. We're actually going to reduce or add electrons. So we're taking electrons away from our carrier and putting them into our organic compound, and that's what helps us form G3P. G3P actually sounds familiar because it was one of our intermediates during glycolysis. Immediately when we broke, we actually formed G3P, and then later ended up with pyruvate. So we see this kind of connection between G3P in glycolysis and G3P in the Calvin cycle. So what have we done? We've added carbon, we've added electrons, and we get G3P. So we see our carbon fixation stage, we see our reduction stage. What is going to kind of be important here is that we notice that it takes 3 turns of the Calvin cycle to get 1 G3P. Well, that makes sense. G3P is a 3-carbon sugar. But what did not happen is that these 3 carbons got put together to form a G3P. We were adding 1 carbon on to these intermediates. So that's a really important thing to notice. The final step is actually the regeneration of RUBP, why it's called the Calvin cycle, and we do need ATP again. So what we see is that we have a higher need for ATP than we do for NADPH, and that is actually why we have cyclic photophosphorylation to make more ATP compared to NADPH.